Hello everybody and welcome to the Carlam Cymru revision sessions. This session this evening will focus on genetic profiling and genetic modification at GCSE level and will be presented by Abigail Kavanagh from Connors Quay High School. The session will last around 45 minutes where Mrs Kavanagh will run through the relevant content. If you have any questions during the session, please use the question and answer or Q&A section and we'll do our best to answer you. You will see that there's a hyperlink in the Q&A section. If you're happy to leave your name and email address, we would love to keep in touch with you so that we can send you information about future events. You can click on the link at any time during the session. Today's session will be recorded and the recording and any relevant resources will be uploaded to the e-sgol website under the Carlam Cymru tab. Thank you, Mrs Kavanagh. Over to you.
Thank you very much, Mr Cobb. Hello everybody and welcome to this GCSE revision session. In this first session we're going to look at genetic profiling and genetic modification. First of all though, we're going to have a look through some things that you should remember to do with DNA. This section of work is from DNA and inheritance in year 11, so you should be familiar with these terms. So let's have a look. So we've got 3 questions here and 3 possible responses. So you could now take this opportunity to pause the video here so that you can have a look at the questions and jot down your answers and then restart again when you are ready. But we will just carry on now and look at the answers together with these questions. Okay, so first of all, what is the shape of DNA molecules? So we've got 3 possible answers here. We've got a base pair, a double helix, and we've got cytosine. And our second question, what is the base complementary to guanine? And again, we've got those 3 answers. And then finally, we've got what is the name of 2 complementary bases held together by hydrogen bonds, either AT or C and G. So let's have a look at our answers here. So the 2 complementary bases held together are base pairs. And as you should remember, we've got adenine and thymine and cytosine and guanine. And then that will help us with our final question in a second. The shape of the DNA molecule is called a double helix. That is an exam question that I've seen in the past, so make sure that you remember that terminology, double helix. And then thirdly, the base complementary to guanine is cytosine. Okay. Let's move on then and look at what we are going to be looking at in this session. So we're going to be looking at genetic profiling and see the stages involved in carrying out a genetic profile, to think about some of the reasons why genetic profiles may be used, and then think about some of the issues surrounding genetic profiles. So for you to be successful then in this session, you will describe the stages involved in carrying out a genetic profile, identify reasons why they may be used, and state some of the issues surrounding that. And our keywords for today, we've got profiles, electrophoresis, criminal, and paternity. Okay, so have a think then now, just based on that information that we've just had a very, very brief look at, and think about why we might use genetic profiling. So pause the film and have a think why we might use genetic profiling. Okay, so some reasons perhaps why we might use genetic profiling. So we might use it when we are, let's see if we can get this right in nicely, identifying a species. Okay, so that's one reason why we might use it, to identify a species. So if a new species has been discovered by some scientists out in the field, they could use genetic profiling, they could compare the DNA of that species with other species that are alive now to see which family group it belongs to. So it can be used in classification. It can be also used, genetic profiling, in paternity testing, to see whether somebody is the father of a potential child. So that is a very important use of genetic profiling. Okay, so paternity testing is our second possible use. Very good writing there. And then another potential use of genetic profiling. So we've got identification of species, classification, paternity testing, and possibly to identify any diseases that somebody might have inherited. There we are. Okay, so we'll move on and look at some of these areas in a little bit more detail. Okay, so the background really as to what genetic profiling is. As you can see here in that image, we've got all of our bases and we've got our double helix shape there. And for everybody that's a human, our DNA has this same structure where we've got the sugar phosphate backbone. Let me get my highlighter and show you. So here we've got the sugar phosphate backbone. And then in the central part of that helix, we've got our bases. And it's the arrangement of the bases that enables amino acids and proteins to form. So we have the same structure of DNA, but there are some differences. And it's the arrangement of those base pairs in the different sequences that enable the formation of amino acids and proteins that makes us different. And it's that difference, which is roughly 1%, that we can use that to identify differences between people in particular situations. And genetic profiling uses sequences here. It is sometimes used as, or known as, DNA profiling, and it was used as early as 1987. Okay, so this image here shows us some scenes of crimes officers, and they are looking for evidence. So there's been some sort of situation here. What evidence might they be looking for? So let's have a think. You can see here on the ground that there are various numbers that have been placed where they would be collecting evidence. Now, once that evidence has been collected, we would then look at that evidence and try and interpret it and use our scientific information, our genetic profiling, to try and help solve a crime. And during that process of genetic profiling, this particular image or an image similar to this would be produced. And what you can see here is very clearly the DNA and the genetic profile banding that has been found at the crime scene. And then we've got 3 potential suspects. And you can clearly see that the banding from the crime scene very clearly matches that of suspect 2. Whereas you can see that there may be some similarities in suspect 1 and suspect 3, but they are not an exact match. And that is the idea behind genetic profiling as well, that it helps to create a very close match between either a suspect or between different species to enable them to match up. So let's look at the process that takes place. So quite simply then, a sample is taken, whether it is something that's left behind at the crime scene, so it could be a blood sample from some broken glass, for example, or it could be a sperm sample, or it could be a DNA sample from a living organism. Enzymes are used to cut that sample up, and the lengths of that DNA are arranged in order. They're clearly labelled, and then the DNA is inserted into a gel-like substance and an electric current is passed through it. And this is that idea of gel electrophoresis, that the electricity, as the current flows through the gel, it pulls the banding of the DNA apart at its different masses, so we end up with our characteristic banding. So make sure that you know then the different uses of that. So the paternity of a child, the family connection, to solve crime, and finally to determine species. So in classification, that's really very important. Okay, and these are some of the jobs that people might use genetic profiling in. So just to run through, biologists and zoologists, perhaps working with animals or plants, as in a horticulturist as well, might want to look at organisms and map their genetic profiles to see how closely related they are to each other to enable their use for various projects. Epidemiologists would be looking at perhaps using genetic profiling to study diseases and how they affect people, and research scientists also would be using profiling for that purpose too. Okay, here are a couple of example questions that you might want to have a think about and pause the film and have a go at attempting them. So the first one is an example of a grade B/C type question, and it's quite a straightforward question in terms of it's asking us to give a definition of DNA profiling and explain what genetic profiling can be used for. So in that case, you would give your definition of genetic profiling or DNA profiling, and then think about some examples that we've mentioned already, what that can be used for. So genetic profiling is using and producing a genetic profile of an organism, and it can be used to determine the paternity of a child, or to help solve crime, or to classify a living organism. And if we look at the AA star type of question, then we need to write a set of instructions on how to produce a genetic profile, and that would be a step-by-step set of instructions based on what we looked at previously, where you would have your sample taken, and then you would use enzymes to break that up, hence using the keyword there, to break up the DNA into different lengths. The lengths are then arranged in order of mass, and then that DNA substance is then injected into the gel, and the electric current runs through and produces that banding that we are familiar with. Now, as I mentioned at the start of the lesson, there are ethical issues surrounding the use of DNA profiling. So I just want you to pause the film for a second and have a think, what might those ethical issues be? Okay, let's have a look then at what those ethical issues might be and why you need to be aware of them, because if you have a 6-mark question perhaps, or a 4-mark question, you might be asked to look at the pros and cons of genetic profiling, so looking at the benefits of genetic profiling, but also looking at the other side where it may be an issue. So firstly, we're thinking here that DNA tests are kept even if somebody is innocent. So in a crime situation, if somebody perhaps is arrested, their DNA is taken, they may be innocent, but that DNA information is stored. So there are advantages and disadvantages to this particular point. So if the information is stored, firstly, do we know where it is stored and how it is stored, and who has access to that information? So here you can see that the ethical issue of ownership might arise because the police keep the profiles on police record. So who owns that information, even though it's your DNA? Do the police own it, or do you own it? It is, though, advantageous that that information may be kept. And you can see here that cold cases from previous years have been solved with DNA samples that have recently been taken. Because that information has been kept on file from a great number of years ago. So there is a benefit, there is a disadvantage to that, and it's just about you putting forward a balanced argument to make sure that you get your 6 points on that particular. So here's another aspect to think about, that NHS Wales would suggest that early warning of rare genetic diseases. So that's our information and our starting point. So the advantage of that is that the correct medical treatments could be given to the patient at an early stage, and any help with treatment. So it would be very beneficial to that person who had that genetic disease that treatment could be started very quickly. But the disadvantage of early warnings would be for insurance purposes, and people may not be given life insurance if they are carrying a genetic disease. So that's the disadvantage for that particular case. Okay, let's just have a look then at something just to finish off with this particular section. Okay, so here is a 6-mark question that has been on an exam paper previously, and let's just have a look at it and what we might want to include in it. So it says, explain what is meant by genetic profiling, include its uses and the advantages and disadvantages associated with it. Okay, so if we have a look then with our highlighter on what's the important information here. So we've got to explain what is meant by genetic profiling. So that's not just giving our definition, but giving an explanation of it as well. And then we've got to include its uses, advantages and disadvantages associated with it. So that means we need to talk about all of those things that we've discussed so far in this presentation. So what is genetic profiling? What is it used for? And what are the advantages and disadvantages associated with it? So now would be a good opportunity for you to pause the film and then to have a look through what your particular answer would be, how you might structure that question, and then restart the film when you are ready. Okay, so here's our information then to help us answer that question. And what you could do is just go through the answer that you've written now and see where you would award yourself points. Remember, you do get 1 mark for the quality of your written communication, so make sure that you have spelt keywords in particular correctly. Okay, so the first part then of the question asked us what genetic profiling was, and here is our particular response to that. So genetic profiling is when DNA is analysed to show a particular pattern, say, or a particular banding that is specific to an individual. So the DNA is cut into smaller pieces. You could include there by enzymes and separated by electrophoresis or gel electrophoresis. That's the name of that process. The pattern that is produced is compared to other samples. So like we looked at towards the start of this session, where we could see the suspect's DNA banding and the 3 other banding compared to what had been found at the crime scene, then that's what would happen next. And then here are a number of different uses. So you would include those uses within the body of your writing. So determine the paternity of a child, to help solve criminal cases, to look at family connections. That's just 3 different examples. And then we could look at the advantages. So the advantages of genetic profiling would include matching parents to children, genetic screening, and providing evidence for criminal cases. And then finally, our disadvantages. So we mentioned earlier about not being able to get insurance, which could be a problem for people who want to buy a house. They may not be able to obtain a mortgage. There may be some inaccuracy in the process. It certainly may be an invasion of privacy if that DNA is stored somewhere. And also, what we've not mentioned so far is that it might affect somebody's chances of getting a job. If an employer knows that you have a potential genetic illness that may cause you to have time off work, then they may not want to employ you. Okay, so that's the first part of our session today, where we've looked at genetic profiling. So we're going to move on slightly now and look at genetically modified organisms. Okay, so there are a number of different keywords here that we will be referring to in this session. So genetically modified, so when we alter the genes of an organism, and advantages and disadvantages. We'll be looking at some case studies of those during this session. Transferring of genes, we'll be discussing that as well, and genetic manipulation, and you can see there genetic engineering, all different terms that we use to describe what happens during genetic modification. Some of this can be used to increase yield and resistance, particularly in crops. And then finally, we'll look at that term transgenic in relation to genetic modification. Okay, so we're going to end up then with a knowledge of the definition of genetic modification, and then thinking of and explaining the advantages of modification and the disadvantages and any potential issues with genetic modification. So success criteria at the end of this session, you will know a definition of the concept of genetic modification, and you will be able to understand and explain the advantages and the disadvantages with any potential issues of genetic modification. Okay, so let's have a think now. We're in an imaginary country in Lilliput, and there's a problem with their wheat and corn yields because of too little rain and locust swarms eating their crops. So what I'd like you to think about is to suggest some types of genetic modifications that we might make to these two crops, so that the people from Lilliput can have excellent crop yields that they are used to ensure everyone is well fed. So let's have a look at the key points that we need to think about here. So there is a problem with our wheat. Oops. Highlighter. Thank you. Right. There's a problem with our wheat and corn yield. Now the yield is the amount of crop that we get there. And the reasons for that is that we've got not very much rain, and we've got locust swarms that are eating the crops. So we need to think about what genetic modifications we could do these crops to make sure that everyone is well fed. So pause the film and have a look and think about what the potential problems are and what the solutions might be, and then think about what suggestions you could make. So pause the film and write down the potential problems that we've got from that particular scenario, what solutions you can think of, and also any suggestions that we might make. Okay, let's have a look then. So we know that the wheat and the corn yield are not what they should be because there is not enough rain and the locusts are eating the crops. So first of all, let's think about what we could do if there's not enough rain. If we were to genetically modify our crops, what could we add into the wheat and the corn to make them survive in dry conditions? So if we think about plants that live in dry conditions that we know already, so the cacti, then you might be able to find a particular gene in there that enables it to survive in its dry and arid environment and implant it and insert it into the wheat and the corn. Similarly, with the locust swarms eating the crops, and we're going to look at an example of that shortly, we could find a gene in another plant that the locusts don't like eating. So there must be a taste or a texture that stops them from eating that particular plant, and we could insert that into the wheat and corn. So genetic modification is about thinking about possible scenarios and possible situations that we know and what we could use to change potential organisms. Okay, so here's our definition then of genetic modification. Sometimes you might see it called genetic engineering or genetic manipulation. And the idea of genetic modification is that it takes the gene from one species and implants them within its own species and also across different species as well. And there are examples that you may have seen and heard of where a glow-in-the-dark gene has been taken from a jellyfish, for example, and inserted into a rabbit, and that rabbit will glow in the dark. So there's a lot of situations where genetic modification has taken place, but there are also some situations where this could be very beneficial to us and our environment. So these are the stages then in genetic modification. So first of all, we select our desired characteristic. So if we think about our starter, our question that we thought about at the start, so we might want to find in that situation the characteristic that enables our plants to survive in very dry conditions. Stage 2 would be to isolate that gene responsible for that characteristic. So we find the gene within the genome and we make a note of that. We then take that gene out and put it into our crop that we want to be able to survive in very dry conditions. And then the idea is then that as that organism that has been genetically modified, as that replicates, then it takes with it that new gene. And so it is beneficial. And that organism that has had the genetic modification done to it is called a transgenic organism. Okay, so we're going to look at 3 different case studies now of different types of genetic modification that's taken place. So we've got the Protato, the Venomous Cabbage, and Flavour Savour tomatoes. So what I'd like you to do is just take a copy of this table. So we're going to look at what the modifications are, or the changes that have been made to each of these 3 case studies, and then think about the benefits of each of these changes. So let's have a look at the Protato. So as the name might suggest, the Protato isn't just a potato. It contains protein as well. And this potato has been genetically modified, and it's been genetically modified with some extra protein into it from the amaranth. And by doing so, scientists have increased its yield and increased the size of the potato from between 30 to 50% increased yield. So not only are people getting the carbohydrate from the potato or the Protato, they are also getting a source of protein as well. And this Protato has been tested on people around the world, as well as on animals, and there are no side effects from this change to the genetic information in that Protato. So this is one of the benefits then of this. So if we're looking at this in terms of filling in our table, the modification that has taken place is to enable it to have more protein and to have a larger yield. And the benefits of that are there's no side effects when anybody eats it. It can support the poorest people by giving them a source of carbohydrate and of protein. And that's very important for people in developing countries because they've got the protein and the carbohydrate, and that would prevent them from getting any deficiency diseases. So let's look at our second example, the venomous cabbage. Now, as that name suggests, the venomous cabbage contains a venom or a poison stick to caterpillars. And it's actually derived from a scorpion and from the scorpion sting. And there are a number of benefits and a possible disadvantage really, but we'll talk about that shortly. So the venomous cabbage has been genetically modified to include venom in its genes from the poison of another organism. It is poisonous to caterpillars, and that means that when a caterpillar eats or tries to eat the cabbage, it won't survive. There are a number of benefits really here because farmers won't need to use a pesticide to spray on the cabbage. So economically, this is a good thing for the farmer. So they won't need to use a pesticide, so the time and the money that would be spent on that doesn't need to take place. The cabbage is human safe, and obviously then the effects that chemical sprays might have on the cabbage doesn't happen, and also doesn't affect our ecosystems. There is one problem though with this, is that if the caterpillars do eat the cabbage, they are no longer part then of that food chain, and that can lead to an impact on biodiversity, and that's something that you might need to consider if we have to look at the benefits and the drawbacks of genetic modification. Our third example here, which are flavour saver tomatoes, and this was the first genetically modified tomato that was produced, and it was altered so that it had a longer shelf life, and it was developed so that it could be transported very easily, and it didn't get bruised and it didn't get damaged by transporting it on a longer distance. Safe for human consumption, again, because it had a longer shelf life, it wasn't sprayed in the supermarket to keep it fresh. But unfortunately, this tomato isn't made anymore because it was very expensive to produce. So a couple of things really that I want you to think about, just as an aside, while you are thinking about this topic really. Was it ethical to test the Protato on animals? These are just points to consider. Secondly, what effect might the toxin in the cabbage have on the biodiversity in that particular area? So if the caterpillars eat the cabbage and then another organism would come and eat the caterpillar, but the caterpillars are no longer around, then what happens to those organisms that would eat the caterpillars? Can they find another source? Does it affect that food chain? And then in terms of biodiversity, would we support a very big community of organisms there? That's just things to think about. And then finally, because the tomato is no longer produced because it was too expensive, should cost be a factor in this when we're growing food? It might be something that we could do to support people who need food and can't afford to get it. So should cost be a factor in genetic modification? Okay, so a few true or false questions here to finish off with. So I just want you to write down what you think to these answers. Just put true or false. So GM crops are expensive to develop. Is that true or false? They are very expensive to develop. And there we saw that the flavour saver tomatoes are not produced anymore because they are too expensive. GM crops pose no danger to natural species. Again, true or false? Write down what you think. It's false. That there are some situations, and scientists are still investigating whether there is a mixing of genes and whether that causes a problem to native species. Our next question, GM crops could solve food shortages. True or false? That is true. And let's just have a look at this question here. So it says, wheat crops may become infested with weeds. Scientists are developing genetically modified strains of wheat which resist the action of herbicides. Evaluate the possible advantages and disadvantages of developing genetically modified herbicide-resistant crops. So there's quite a few key words here that we need to think about in this question. So the information here that it tells us is wheat crops may become infested with weeds. And scientists are developing genetically modified strains of wheat which will resist the action of herbicides. And then what we've got to do is evaluate advantages and disadvantages of developing, quite a long sentence here, genetically modified herbicide resistant crops. Okay, so the idea is then that wheat crops are overtaken with weeds, and there is a great deal of competition that takes place. So if farmers spray herbicides, then not only are they attacking the weeds, but they may well affect the wheat as well. So what we need to think about here are the advantages and disadvantages of developing those types of crops. Okay, so now would be a good idea to pause the film, write down your ideas, your advantages and disadvantages, and then evaluate them. So you need to summarise those key points. Let's have a look now then at what our possible answer might be and what we might include. So the benefits would be to improve the crop yield or the crop quality, and that is certainly important in developing countries. If you introduced herbicide resistance, that would result in less herbicides being used. Weeds would then be quickly and selectively killed, and that's a key point there, that they are selectively killed and not everything is killed. The genes would be, the selected gene that we wanted would be transferred into other species. However, there's your evaluation. Some people believe it's not ethical to interfere with nature in this way. But some people believe that we shouldn't alter what is and has been developed over thousands of years. If the crop has been altered, it could cause allergic reactions in people. So that's something that would need to be tested and evaluated before any crops were released onto public sale. And the pollen produced by the plant could be toxic. So that's something that scientists would need to be aware of before they sold the plants and sold the crops, because it could be toxic. And then as those bees are moving around, it could affect the bees and also could be passed on to humans as well. So there's a lot of things that scientists would need to do to make sure that they are doing the right thing before anything enters into the food chain. Very much for this evening.